as an introduction and an explanation, my name is Joe Hacia. I'm on the faculty of the University of Southern California. I apologize if my voice is kind of ragged right now. I was going to attend the ULF meeting, but I've had a pretty bad cold or lung infection the last couple of days. So I sound worse than what I feel. Hopefully you won't feel that worse after I give this talk today. But I'm going to give, I think, like on the order of 2 presentations. But the first one that I'm going to be talking about is a little bit of a newer topic, which may not fit into this direct agenda that Christy mentioned, where she was talking about emerging therapies and going over a whole bunch of different ideas. I'm going to be going over something called liver-based editing, lessons from Zellweger spectrum disorder and implications for adult Refsum disease. And here it's going to be the liver is going to be the central theme here about why we're talking about trying to address liver functions for a disease that has CNS and visual manifestations, okay? So the objectives, which I hope can set the stage for the other talks, would be to review the central role of the liver in clearing circulating phytanic acid, and then to demonstrate how these liver-based editing, or this gene therapy, restores peroxisomal lipid metabolism and can inform therapeutic strategies for a related disease called Zellweger spectrum disorder, as well as an adult Refsum disease. And then discuss some opportunities of global DARE to accelerate gene therapy research and clinical translation in the longer term, realizing that for this particular conference, we may be more focused on drugs in a short term, like small molecules. But this is that'll be my overview. This is my brother Frank, who I tried to acknowledge in all my talks over the last year. He passed away about a year ago, and he was the person who was my inspiration for going into a field of science, and also my inspiration to try to become a better human being in my life. So I always want to dedicate everything to Frank. As an overview, adult Refsum disease is a rare metabolic leukodystrophy. It's ultra rare, about 1 in a million or 1 in a few 100,000 in various populations. Most of the time it's caused by, it's recessive and caused by loss of function variants in the Phy-H gene, both copies. And this results in the impaired catabolism of a dietary branched chain fatty acid called phytanic acid. And the accepted hypothesis is that the accumulation of toxic stores of phytanic acid in various tissues is causally responsible for disease. And so we see the manifestations from retinitis pigmentosa, hearing loss, loss of smell, as well as ataxia being amongst the manifestations of disease. So humans only obtain phytanic acid from dietary sources, and most of this comes from ingestion of ruminant fats, and they're getting this phytanic acid from the ingestion of chlorophyll. And so those animals, such as the meat of a cow, can be very, and the fat from that animal may be very enriched for phytanic acid. And so humans can't generate it by themselves. They have to ingest it. And people on what so-called Western diets generally ingest about 50 to 100 milligrams of phytanic acid daily. So when you think about how this fat is tracking in your body, when you ingest, something like a hamburger, and you have your dietary lipids, it's going to be going through your system. And the phytanic acid itself is going to not just be in the free form, but be esterified or attached to many other different types of molecules. It could be attached to cholesterol, it could be attached in membrane lipids. It could also be stored in triglycerols or triglycerides. And it's during this digestion process, especially through the enzymes secreted by the pancreas, that the phytanic acid is liberated from these stores and eventually then repackaged in intestines into molecules called chylomicrons. And here's an example of a healthy intestine where producing these chylomicrons after a meal, and it's going to be carried through the lymphatic system in the beginning and secreted through the lymphatic system. Eventually it gets into circulation, and then it's going to be able to deposit these, or carry, I should say, these fatty acids such as phytanic acid and other fatty acids to peripheral tissues such as adipose tissue. And then over time, as this chylomicron gets metabolized in the body, it gets into smaller particles called these remnants that get taken up by the liver and further metabolized. The phytanic acid, which has been deposited in the adipose in the form of triglycerides, can also be liberated in times of stress, where the phytanic acid can be liberated through lipases, get into circulation, and again, it's the liver that is playing the major role in turning over this phytanic acid and preventing it from accumulating in very high levels in the body. So therefore, that's why I'm saying that the liver is the metabolic hub for fatty acid metabolism in both the fed and fasting states. So there's a couple of different ways, if you start looking within the liver, about how phytanic acid is broken down. And it's broken down in the cells in the liver called hepatocytes. The main pathway is called alpha-oxidation, which accounts for the bulk of phytanic acid catabolism, where you start out with here's phytanic acid, and if the process goes to completion, you produce carbon dioxide and water. There's, in most people with adult Refsum disease, there's a defect in this pathway. In fact, in the gene called Phy-H, which encodes an enzyme called phytanyl-CoA hydroxylase. And this is a key enzyme in the metabolism of phytanic acid. And if you have defects in this particular enzyme, or the amount of it, or activity, that's when you start accumulating phytanic acid. As we will talk about more later, and it'll be the star of the show for another presentation, omega-oxidation is an alternative way of breaking down phytanic acid, and it accounts for like a fraction of what alpha-oxidation is able to break down. But nevertheless, this sort of backup pathway is very interesting to us because there may be ways of trying to stimulate this backup pathway, which is still active in people with adult Refsum disease. And if we could activate that, then perhaps we can use that as a way to leverage that to try to reduce phytanic acid levels that are circulating in the body of a person with adult Refsum disease. Okay, so I will spare you all the chemical steps and go back to more genetics, which is basically my playground. I'm more of a geneticist by training. So I'm going to be talking a little bit about gene therapies. And we know that in people with adult Refsum disease, there's going to be genetic misspellings in this Phy-H gene the vast majority of the time. And wouldn't it be wonderful if there was a technology where you could take that misspelling of an A, C, G, or T that's inappropriately now a different letter, and be able to restore it to its normal state so that the gene is active. And gene editing is an emerging reality, especially for diseases with impaired liver function. And that's why I want to emphasize in this prelude about the real importance of addressing liver function in adult Refsum disease. It was in 2017 that a seminal paper came out of the lab of David Liu. And David Liu is a chemist over at Harvard University. And he was able to generate this base editor system based on the CRISPR-Cas9 platform. I'm not going to go into the details of this because it's a mixed audience, and forgive me if I'm not going over every step in the process. But on the other hand, you don't want to listen to my crackly voice for over an hour anyway. So the idea is that he was able to generate a guided DNA repair enzyme. So this base editor is able to correct genetic mistakes in a very targeted manner. And the way that it's able to target it is by taking a protein that is able to do the repair and be able to use as a GPS signal, as I think of it, a small piece of RNA that binds to the DNA region of interest and be able to cause DNA misspellings to be repaired. That's the big picture of it. So in about 1 year ago, there was a big news story that was across the internet, not quite as popular as a cat playing with some yarn, but nevertheless, in our science world, we were pretty excited about it. And it talked about a patient who had the first undergo a personalized gene editing treatment for a liver disease. And the child's name was baby KJ. This was published in the New England Journal of Medicine in 2025. The people that I've underlined are people that I've either worked with or conversed with quite a bit in this field. So to kind of give you an idea about why people were excited about this, and there was a 5-month-old boy, KJ, who was born with a fatal genetic disorder caused by liver malfunction. It's called the urea cycle disorder. And this young boy has an inability to process, or an impaired ability to process nitrogen in his body. Normally, you secrete excess nitrogen in the form of urea in your urine. But in a boy like KJ with this genetic disease, it would just accumulate as an ammonia in his bloodstream, and as a result, cause severe brain damage and fairly sudden death. So at this particular case, there was just one gene that needed to be repaired, and the gene, really the target organ, was the liver. And they went from birth to month 8 to get a treatment for this young boy enabled. So not only was he diagnosed very quickly, but they were able to get this technology off the shelf and treated this child. So the idea was that they infused him with this base editor in the form of a lipid nanoparticle. So the lipid nanoparticle is very similar in theory to the delivery system of mRNA for the COVID vaccines. You have an mRNA inside here which encodes the base editor. You have a small piece of RNA that tells the base editor where to go in the genome. And then you infuse the person with this particular drug, and eventually it gets taken up in the liver cells, and then it could do its magic of trying to repair misspellings in the gene of interest. So if you want to look at the data behind this, and I'm going to go into the data because I think it's important to kind of understand what this type of technology can and cannot deliver, and how it relates in broad sense to adult refsum disease. So in adult refsum disease, you want to prevent spikes in the compound called phytanic acid. And for baby KJ, you want to prevent spikes in ammonia that's circulating in his bloodstream. And any spike in ammonia that goes beyond a certain level can be fatal to him. So this is a graph showing the levels of ammonia in his bloodstream by age 3 months. This is before treatment. Every one of these dots is a measurement of ammonia in his bloodstream. Anything below the dotted line is tolerable. Anything above the dotted line can potentially be fatal to him if it was for a prolonged period of time. So you can see that most of the time he's below the line, but he could have these spikes. So they gave him a couple of doses of this drug. And then over time, it didn't prevent the spikes completely, but it dramatically reduced their magnitude. And there was also a compound that was present in his urine that you would normally say if the drug was working, it should actually start spiking up. So here's this baseline, it's very low. If the drug is working and causing that gene to be active, this compound should be higher in urine, and you're seeing this is occurring. It's not a complete fix, but it's something that is actually quite remarkable. So in March 2026, here's a picture of KJ and his mom, Dr. Karen Musinero, over at UPenn, who was one of the physicians, along with Dr. Rebecca Ahrens-Nicholas, the other physician who were the leads on this study. And there they are interviewing KJ, who is looking pretty happy at this particular point in time. So it made a lot of press, but what I really was really impressed with by Dr. Musinero is that both him and Dr. Ahrens-Nicholas have won many awards for this work. In fact, it was touted in the New York Times as being the greatest advance in biomedical research in the last 10 years. So he did say, to be brutally honest, there's profound limitations. There's a lot of enthusiasm and excitement, but it wasn't a clinical trial. It was just one person, and it was not a cure. He's saying that we could turn a devastating disease into a milder, manageable condition. So it was a personalized therapy, and we can't be snake oil salesmen and give false hope. And that's really important to me when I discuss any of the therapies on the docket today, to not oversell them, to have cautious enthusiasm, but to give you the full story as much as possible. But nevertheless, this is still a breakthrough in my particular opinion, and it has some implications for adult refsum disease, as I will now start to talk about. So there's a lot of rare genetic diseases where the liver activity is missing, liver is missing a protein that's critical for human health, and adult refsum disease falls into this particular category. So as such, I'm very excited that, by banding together with the larger rare disease community, that we can be able to try to get these types of therapies out into the clinic. Now, this is a paper I just published with some colleagues in Nature Biomedical Engineering in 2026, looking at base editing to correct a liver disease in a disease that's actually highly related to adult refsum disease. It's called Zellweger spectrum disorder. And many of the authors on this particular paper were also involved in the baby KJ story. So Zellweger spectrum disorder is a related disease, but it's generally more severe than adult refsum disease, because not only do these kids have an impaired ability to break down phytanic acid and develop stores of phytanic acid if they live long enough, but there's a lot of other metabolic issues that are affecting their health. So there's a commonality of the phytanic acid thread, but this particular disease is generally more severe, and some of the children can, on the more severe end, live 1 to 2 years of age. For the adults with the disease, can live a longer lifespan, but many of the kids on the moderate end of the scale or middle part of the scale will have developed severe liver disease. So instead of Phi-H being the gene of interest, they're all genes called PEX genes. There's one specific misspelling called G843D. It has to do with amino acid substitution in this particular protein. And it's about 30% of the patients have this specific misspelling. So it was back in 2019 that I contacted David Liu about this and brought a team of investigators, not only from Harvard, but the Jackson Laboratory, Kennedy Krieger Institute, and McGill. And here's me, not to brag, but I won the best Easter sweater award at a swing dance. So I won a chocolate bunny. So I'm a very well-decorated researcher. The idea here is that it was a multi-year process. And it was a lot of, all the work was really done in Maine at the Jackson Laboratory and at the Broad Institute at Harvard. I want to give credit where credit is due. So we took this mouse, which has, it develops a very severe liver disease as a result of having a mutation in the SPEX1 gene. And what we do, or when I say we, it's all Kat Lutz's group over at the Jackson Labs, is inject the magic base editor, which is delivered through a virus, an adeno-associated virus, in the facial vein. And we look at the amount of editing that will occur after 6 and 16 weeks post-injection. And at the end of the day, at the high dose of the editor, we're getting about 60% of the cells in the liver have complete repair of the mutation. And in fact, it's about 60% of liver cells are hepatocytes. So we predict that just about every hepatocyte in that mouse liver has been repaired. When we go to lower levels of the virus or the drug, we get about 30% repair at 6 weeks, but then it pops up at 16 weeks to just about 60%. And it implies, but does not prove, that the cells that got corrected have some sort of survival advantage. So what's interesting to us is the fact that when you look at these mice, they tend to be smaller in general relative to mice that are healthy. So if you look at age in weeks and body weight in the males, here in black is a wild type animal that is growing at this growth trajectory in black. In blue, this is one that isn't treated with the base editor, so you can see it's smaller. And then when it's treated with the base editor, it actually rescues the growth. And the exact same thing happens with the females. So that, excites us that the animals are growing bigger. And then we started looking at the levels of peroxisome metabolites. So the ones that, as I mentioned, Zellweger spectrum disorder is marked by an impaired ability to break down Phytanic and pristanic acid. And this is these branched chain fatty acids. And in black here is normal levels of pristanic and Phytanic. This in red, this is from an animal that has the mutation. And this is a post-treatment animal. So you can see that by correcting the liver, we could reduce Phytanic acid levels back to wild type. And in fact, there's other types of fats that, very long chain fatty acids, that are also corrected. So it's showing that when we correct the liver, though, we can correct the branched chain fatty acid levels. And in these animals, they get fatty liver disease. So a section of a liver stained with oil red O, which looks at lipid droplets that will accumulate as part of the pathology. Anything with the angry red dots over here shows an accumulation of lipid droplets. This is the untreated animal in the middle. Treated on the right-hand side, you could see a dramatic diminution of these lipid droplets. So that excited us. Now, from the perspective of adult Refsum disease, we also took mice that were injected at various time points, and we looked at the levels of Phytanic acid and pristanic acid, as well as a number of other fatty acids in the brain. All that work measurements was done by Ann Mosier, who was very spectacular in what she has achieved for this project and everything else in her storied career. So when you look at this, what we call a volcano plot, the idea is that anything in this upper right-hand corner is going to be an elevated fat. So we see elevated Phytanic and pristanic at 6 and 16 weeks in untreated mice. But when we treat them with the base editor, the levels of this Phytanic acid are no longer high in the cortex of the brain cortex of these animals. And the idea generally is that by reducing the circulating levels of this Phytanic acid, you're preventing it from accumulating in the brain. So fixing the liver can have a dramatic effect on the peripheral tissues. Hence why we're focusing in on the liver for now. This can be done at animals that are treated like a month after birth. The exact same thing can occur, that before and after we're seeing Phytanic and pristanic acid being elevated, but now after treatment, it is now back to normal levels. And then we've also been able to deliver this base editor using these lipid nanoparticles, and we see something very similar, that we can correct the phytanic acid levels back to normal after treating with the base editor with the lipid nanoparticle. So our hope for Zellweger spectrum disorder, and ultimately, in my opinion, for adult refsum disease, but further in the future, is to be able to develop these lipid nanoparticles where we could deliver the base editor and try to repair the mutation in the livers of people with these diseases, so that we can be able to restore phytanic acid oxidation and reduce the body burden of phytanic acid throughout the body. All right. So what was very inspiring to me was that after the paper was, our paper was published, there was a press release. And David Liu, the lead investigator at Harvard who invented this technology, had a quote. And he said that when he was approached by Kieran Musunuru at UPenn about baby KJ and what version of the base editor to use, he relied upon our data from the PEX1 project to be able to select that base editor. And he said it's easy to assume that rescuing this quite rare peroxisome disorder in mice wouldn't have anything to do with saving a human with a very different life-threatening disease, but it had a lot to do with it. And so the spirit here is that although I've been talking about PEX1 and Zellweger, it had an influence on baby KJ's life, I hope. And it could also have an influence on our discussion about the treatment of adult refsum disease. But there's a lot of caveats that we will get into. That, it doesn't work for every genetic glitch. The cost estimates are very, very high right now, $1 to $2 million per patient treated. Demand is high, access is limited, and there's some safety concerns. So there's a whole genetic toolbox, which I will not get into, about how you could be able to repair mutations in DNA. I wish it was just the base editor, but no, there's different tools in the toolbox to be able to address different types of mutations. For the adult refsum disease community, it probably would be this prime editor technology, but it does essentially the same thing of trying to repair the mutation in a person's DNA. But there is also a decision balance that should come into any of the therapies that are under discussion. So there's a risk benefit, and this would be up to the physicians to discuss. I know that I've talked to Florian Eichler and others about this, of a disease like adult refsum disease, which in theory can be managed to a certain extent by diet and by plasmapheresis. It wouldn't be immediately life-threatening, even though that's not to diminish the impact on a person's quality of life and vision and senses, as well as the peripheral neuropathy, etc. But nevertheless, that risk-benefit does come into play. For example, baby KJ could have died if not being treated, versus a person with adult refsum disease can live a long life. So therefore, this risk-benefit analysis comes into play, and it's something we have to think about. For a technology like this gene editor or base editor, we always talk about the happy story is that there's an on-target edit, and we fix the mutation in the DNA, and then we're all happy. But what happens if there's a mistake, and the editor comes in and then just changes a different spelling elsewhere on the chromosome? And it turns out that is pathologic, and then you give somebody another disease. That's something you have to talk about and think about Well, if it's 1 in 1,000 cells that develop a mutation somewhere in your genome or in your DNA, would that be problematic or not? So our goal in this therapeutic editing is to introduce risks that are smaller than ongoing processes present in your tissues. So I did a little bit of homework on this because I wanted to be able to show some data. And it was a very interesting biology, in my opinion. They took, the investigators here isolated hepatocytes, or these cells from the liver of human donors from anywhere from 5 months to 77 years of age. And they just asked a very simple question. They said, do these cells have mutations in them already that they acquired during their lifetime? Or are they, pretty pristine clean? And here is the number of mutations, these single nucleotide variants per cell. And this is times 1,000. So from the liver sample, the cells from someone who was about 1 year of age or so had about 1,000 mutations per cell that was acquired during their lifetime. And people who, here, there's the young and there's the aged. I'm in the aged category. There's an increased number with age. So the idea is that it's normal to acquire mutations in your liver over age. Pretty much almost all cells in your body accumulate these types of mutations. It doesn't always result in something like cancer, but is something to consider. So if you have a genome editor that only introduced 1 or 2 mutations elsewhere in the genome, you're probably okay given the fact that the baseline is thousands. You just have to make sure that mutation did not inadvertently occur in a very bad place in the genome, like inactivate a gene that would protect you from developing cancer, okay? So I'm just kind of putting this into context that there is biology behind it. The conclusions just for this base editor talk over here, which I recognize is a little bit further afield than the 5-year plan, but our work with this PEX1 mouse model demonstrated that correcting a single gene in hepatocytes in the liver restored peroxisome functions and lowered circulating phytanic acid. So all you have to do is basically hit the liver and you can lower the phytanic acid levels dramatically. And then if you lowered that circulating phytanic acid in plasma, then you could actually reduce its accumulation in other organs such as the brain. So hepatic correction can benefit distant organs. Now, both Phy-H and PEX1 converge on this alpha-oxidation pathway, suggesting that restoring hepatic Phy-H activity could reduce systemic phytanic acid exposure. And by definition, you could also say that restoring or influencing, nudging upward, omega-oxidation pathway in the liver would also rescue phytanic acid exposure to other tissues. So this provides a rationale for this base editing long-term in adult refsum disease. And the next challenge is to consider is translating this biology into safe and durable therapy. So all this work wouldn't be done without a large number of collaborators. It's really at the Jackson Labs where all the mouse work was done. At the Broad Institute at Harvard, that's where David Liu's team did their magic in developing these editors, as well as collaborators and physician scientists at McGill, University of Massachusetts. So, and we were funded through NIH grants and the Global Foundation for Peroxisomal Disorders.